Good morning, and thank you for joining us for Winship's Grand Rounds this morning. If you're an Emory employee, healthcare, or university employee and would like to receive a CME credit hour for attending today, the access code today is 110087 and can be found in the chat feature at the bottom of your screen for our virtual attendees. If you have any questions with this webinar or the CME login, please send Katiatu Fofana an email or drop a note via the chat feature. This morning, we are pleased to welcome Dr. David M. Schuster. Dr. Schuster earned his medical degree from Albany Medical College in Albany, New York, where he completed his internship as well. He completed his residency in diagnostic radiology at Tufts University in Boston, Massachusetts. He conducted his fellowship in nuclear radiology at Emory University here in Atlanta. Dr. Schuster is a professor in the Department of Radiology and Imaging Sciences at Emory University School of Medicine. He also serves as the director of the Division of Nuclear Medicine and Molecular Imaging and is a Georgia Research Alliance Distinguished Scientist. Board certified in both radiology and nuclear medicine, he specializes in molecular medicine and integrative imaging. He is a member of the Discovery and Developmental Therapeutics Research Program at Winship Cancer Institute. He is a fellow of the American College of Radiology and holds professional memberships with the RSNA, ARRS, Society of Nuclear Medicine and Molecular Imaging, and American College of Nuclear Medicine. Dr. Schuster's research focus concerns projects involving molecular and nuclear imaging, with a special emphasis on positron emission tomography, cancer imaging with novel radio tracers for cancer diagnosis and staging, especially the use of amino acid transport imaging for oncologic applications. He is the PI on multiple NIH studies and other sponsored research grants. Please welcome Dr. Schuster today for his talk regarding novel imaging of lobular breast cancer, insights from an R21. Welcome.
Thank you very much. And thank you for having me, and thank you for that introduction. So today we're going to talk about novel imaging of lobular breast cancer and insights from an R21 that was successfully performed at Winship. So invasive lobular breast cancer has been called initially indolent and slowly progressive. There is generally low FDG uptake, so there's limited utility of conventional FDG PET for diagnosing ILC, or invasive lobular cancer. And one of the reasons also is ILC has a discohesive growth pattern. There's loss of EK adherent, which kind of keeps the cancer cells together. So it tends to spread in more infiltrative manner. And there's a metastatic rate of up to 43%. This is a case that we had here where this patient had a distended stomach. And on PET, really not much is lighting up. On CT with contrast, maybe you could appreciate a little better, a little mesenteric stranding, but really nothing to write home about, but it was causing a gastric outlet obstruction. And this was biopsy proven to be invasive lobular cancer, kind of showing how sneaky this disease is. So we have a problem in that no accurate imaging techniques really exist for staging. We're talking more whole body staging of invasive lobular cancer. It's difficult to visualize metastasis with conventional imaging modalities such as CT, bone scan, and MR. And the suboptimal imaging adversely impacts accurate staging upon which therapeutic decisions are based. So our goal with this project was to address an unmet public health need by improved staging of ILC with novel imaging, specifically to detect metastatic disease, which then may inform optimal management decisions. Possible outcomes could be maybe an improvement of health and survival in the nearly 40,000 women who are diagnosed with ILC per year. And at some future point, if we have better imaging, maybe we'll have also a better imaging biomarker of therapy response. So we had some preliminary data, and this was funded with a Glenn Family Breast Center pilot grant from Winship, just to show you how these small projects really kind of come to fruition sometimes. So we imaged 17 lesions in 12 patients, published this in 2016, and we used fluciclovine PET, which is an amino acid PET, which was invented here by Dr. Mark Goodman And many of you know that we use this for prostate cancer. Is actually FDA approved for prostate cancer. But ultimately, amino acid metabolism is really agnostic to many types of cancer. So we could see here that when we're looking at all comers for breast disease, and we really only looked here not at metastasis, but primaries and local regional extent, malignant disease had a greater uptake, and is just kind of over time, than benign disease as well as normal breast. Now, in our preliminary data, there were some interesting things. Most of the patients were invasive ductal, but 5 out of the 13 malignant lesions were invasive lobular cancer. And we noticed that fluciclovine uptake in invasive ductal cancer and ILC were higher than in benign lesions. But even though IDC had a higher uptake than ILC in the primary tumor, in axillary nodal disease, this was flipped. And this is still a little bit of a mystery. ILC nodes have greater uptake than IDC involved nodes. And we could see the mean SUV max was about twice that. Interestingly, uptake in fluciclovine in ILC local regional disease was greater than primary lesion on the average. 2 ILC patients also had FDG PET, and the mean SUV max was higher for fluciclovine than for FDG. A similar study was going on at the same time at Sloan Kettering, actually. 8 out of the 27 patients had ILC in their series. And also they found that there was some utility, but SUV max of axillary nodes in ILC was greater than IDC. And also ILC nodes had greater uptake with fluciclovine than in the primary lesion. 4 patients with ILC in their series underwent FDG and fluciclovine PET, and fluciclovine avidity was greater than FDG avidity. Distant disease, again, was not studied in either trial. This was kind of a preliminary trial of local, regional, and primary disease. So from that series, here we have a patient with multilobulated ILC, left upper outer quadrant. We could see uptake with fluciclovine and really good intense uptake with fluciclovine in an axillary node, which is proven to be metastatic. Also, from this series, here we have the same patient, imaged with fluciclovine and FDG, and for this lesion within the breast, there was greater uptake with fluciclovine than with FDG. We also did a post hoc analysis. We did whole transcriptome RNA sequencing, comparing those lesions with high fluciclovine uptake to low uptake tumors. There weren't a lot of tissue, to do this with, but we found that set-based enrichment analysis revealed significant enrichment of several KEGG pathways with pertinence to cancer biology and metabolism. So this involved PI3K, AKT signaling. We also know that fluciclovine reflects amino acid metabolism, which is associated with the mTOR pathway, HIF1 signaling, cysteine metabolism, central carbon metabolism, and Notch signaling. So kind of sitting around thinking about how can we exploit this exploratory data. And we decided to write an R21. And the intent was to more fully test fluciclovine and ILC, especially for distant disease. But at that time, there was an editorial in the literature, and everyone was going PSMA crazy. And so PSMA is prostate-specific membrane antigen, and it's now FDA approved in a number of forms for evaluation of prostate cancer. But as I have said in some of my other lectures, and some of you have been to those, PSMA is neither all prostate nor entirely specific. So PSMA targeted imaging, they advocated in breast cancer, should really be explored for systemic staging, especially in lobular cancer. So I decided to take them up on that challenge. We submitted an R21 in June of 2020. It was awarded. and this was the project period to late last year. So what's our rationale for fluciclovine? Well, amino acid transporters, especially LAT1 and ASCT2, are overexpressed in breast cancer, and they're associated with tumor growth, metastasis, and hormone receptor status. And correlation of fluciclovine uptake and malignant potential was reported earlier in vitro in breast carcinoma cell lines and in animal models, some of this research being done at Winship. So what's our rationale for PSMA? And this was the radiotracer that was most available to us. Now we have fluorinated versions, but gallium-68 PSMA-11. So PSMA receptor upregulation is noted in tumor-associated neovasculature. So it is non-specific in that way. And this is likely due to PSMA cleavage of vitamin B9. It stimulates oncogenic signaling through glutamate receptors with downstream activation of PI3K, AKT, mTOR signaling pathway. And most data, though, and there's not a lot in the literature, regarding PSMA with breast cancer was pertaining to IDC, invasive ductal. But if we thought if PSMA works with ILC, well, then you could have a theranostic approach that you really couldn't have with fluciclovine, because we have lutetium-177 PSMA radiotherapy. And we're doing that now as Pluvicto in our theranostic center. So our hypothesis was that metabolic imaging with amino acid transport PET will improve the staging of ILC, particularly for distant mets compared to conventional imaging, and that receptor-directed PSMA imaging of tumor-associated neovasculature may complement this metabolic interrogation, reflecting perhaps different features of PI3K, AKT signaling. And to understand the utility of these two PET approaches, we decided to study each in the same patient in a trial, which was also a little daunting in themselves, to try to get cancer patients to sign up for 2 PET scans. But that was one of the reasons why we did the R21, to see if even this type of study is feasible. So our specific aim 1, determine improved detection of METs with fluciclovine and gallium PSMA-11 versus conventional imaging. As confirmed with histology, our goal was to image 20 patients. And if possible, we would biopsy in the safest manner, if not already done, as many lesions as possible. Aim 2 was to determine the concordance and discordance of ILC detection with PSMA versus fluciclovine, and compare the verified detection rates and compare them to conventional imaging. Finally, I put in an exploratory aim. And that's also out of kind of a scientific interest, but it's kind of nice, and from a grantsmanship viewpoint, you just don't want to do a clinical trial and be a lumpologist, as I like to say. You really want to tie this back into the biology. So we wanted to establish the role of molecular detection by ctDNA mutations, including PIK3CA, ESR1, HER2, AKT1, in characterizing the degree of tumor burden, as identified by both of these radiotracers. So we collected peripheral blood. Our plan was to do that in ctDNA in all patients and determine if the molecular tumor burden correlated with the presence or absence of METs, as we found this on the either PSMA and or fluciclovine, the imaging. Our primary endpoint was confirmed detection rate. Secondary endpoint was involving ctDNA for presence or absence of METs and to correlate with the results of imaging. So our inclusion criteria were, and these evolved a little bit over time, but biopsy-proven ILC patients not currently on systemic treatment. At first, we tried to say, well, they've never had systemic treatment. That would turn out to be a little too difficult to accrue. So we just said, okay, they just had to be off systemic treatment for 4 weeks. They had to have either clinical or imaging suspicion of metastatic disease or proven metastatic disease, but there was suspicion of a greater tumor burden that could potentially change the approach and ability and willingness to undergo biopsy if clinically indicated. This was our schema, and I'll run through some of this, but basically we would consent the patient, have a screening period of 30 days, and then within 2 weeks of each other, we would get both PET scans and then do kind of our follow-up assessments and our analysis. Now, we did not want to prescribe what standard of care imaging is. And that's also kind of a practical viewpoint because insurance doesn't always approve, for example, FDG PET, et cetera. So we broadly define this as mammography, ultrasound, bone scanning, FDG PET, CT, and/or MR per usual clinical practice. And it had to be done within 6 months of enrollment, though ideally at 2 months, within 2 months. But again, this was not under the control of the study design. Again, we did PSMA 11 and flucyclovine PET on the same patient. And our goal was to do both in the same week. And we actually mostly succeeded in that. And just to understand some of the physics involved. So gallium-68 has a 68-minute half-life. That means that 10 hours later, it has essentially disappeared for all practical purposes. So you could do gallium PSMA, and then the next day you could do flucyclovine, okay? So that makes that practical. If you're gonna do flucyclovine first, I think we had to do that maybe in one patient. You could do flucyclovine, and then since fluorine-18 has a 110-minute half-life, you really wanna wait 2 days and not do it the next day. So, and then we did the blood draw for the ctDNA, typically at the time of the first PET, to make it easier for the patient. We had a little bit of a complicated interpretation schema, but we wanted to do as much a concordance, discordance between reader analysis as possible. So I and Dr. Musa here blindly interpreted the 20 cases. But of course, when you have a patient, and it's not like you have all these patients at once. So, and we want to try to interpret this in a timely manner. If you look at the first case, let's say, which is a PSMA, you kind of have a hint of what the flucyclovine would look like. So what we did was we alternated. So blinding first 10, let's say I would do flucyclovine and Dr. Musa here would do PSMA, then we would switch that. And then we got the person who was in charge, who ran the trial at Sloan Kettering, who since then went to Hoag in California, Dr. Lehner, to blindly interpret alternating PSMA and flucyclovine. And then this way we would kind of had a good idea of reader agreement. And afterward, an unblinded reconciliation interpretation took place with all 3 readers, just for kind of practical clinical matters. So we divided the patient into kind of 3 regions for metastasis. First, we have ipsilateral axillary lymph nodes. Then we had extra-axillary lymph nodes, which were defined as ipsilateral, supraclavicular, and internal mammary, and then distant disease. And we read these out. We rated them on a 6-point Likert scale. Basically, Likert scores 1 to 3 were benign, 4 to 6 were recorded as malignant. So it's kind of like definitely benign, probably benign, possibly benign. And you know, some of these Likert scales, sometimes we have an equivocal one, but Dr. Krupinski, our research chair, talked me into, or vice chair, talked me into saying, don't do equivocal. Just do it like, end up having binary. I said, okay, we'll try it out. And then we also recorded the actual uptake, the SUV max. And a subset of these lesions were kind of new cases. So we also knew that we could look at intact breast lesions in a subset of patients. So we did that as well. As a reference standard, we had 1-to-1 matching to a composite standard truth. So for example, if we had a region that was positive, we made sure that particular lesion was the one that was biopsied that we call positive. It couldn't be that we called one lesion, let's say in a distant disease negative, and the patient happened to have distant disease we didn't detect, okay? So it had to be 1-to-1 matching. Histology was considered the highest standard of truth. And then we also had clinical and imaging follow-up, like a definitive appearance on correlative imaging in some cases, or interval imaging correlating with clinical progression, or resolution following disease-appropriate treatment, because you can't biopsy everything. And the composite standard of truth is composed of the histologic and the clinical and imaging standard of truth. Any lesion with insufficient standard of truth or determined to be false positive was considered negative. So here are the results. First, we ended up consenting 22 patients, and we fully recruited the study, and thanks to all my colleagues in the breast section of Winship. Now, first lesson learned, there was a big contracting delay. So we started this like 8 months later than we wanted to. So it behooves us all to complain to research leadership about things like infrastructure and contracting. And I think the RAS really has it down now for, and they don't do contracting, for when you put in grants. Okay, I've been very, very pleasantly surprised. Recently, not surprised, but happy with those results. But contracting, I think, is something that we still have problems with. Anyway, we consented 22 patients. 2 patients withdrew for various reasons, and we're allowed to replace those. So we ended up with the 20 patients we wanted. There were no screen failures. Average age was a bit older, though we did have a range. All the patients were ER positive. Usually about 90% of lobular cancer is ER positive, okay? In this case, just all the patients were ER positive. Most were PR positive, only a few were HER2 positive. 7 out of the 20 were undergoing the initial staging, so they had the theoretically primary disease to look at, and 13 out of the 20 had a primary tumor resection of various types. CT and bone scans were done as conventional imaging in 13 patients, FDG PET in 7 patients. And the mean time between conventional imaging and the first research PET was 18.8 days, so that's pretty good. So even in our range, up to 67 days, we kind of kept to that 2 months, which was great. So here are results, and we could see here on first patient and then region and combined region. So the confirmed detection rate, as determined with the standard of truth, was higher with fluciclovine than it was with PSMA or conventional imaging on patient level. And when we combined the various axillary, extra-axillary distant mets, we see that both on the individual region level and the combined, the fluciclovine did better than PSMA or conventional imaging, and various combinations, either including or not including the axillary, for example. And many of these were statistically significant. So when we look at conventional imaging versus fluciclovine, that was significant at the patient level. At the individual region level, the numbers were just really too few, I think, for any significance. But when we combined regions, we could see that not only was fluciclovine better than conventional imaging at the combined region levels of various types, but also fluciclovine was better than gallium PSMA for detecting regional level metastasis. So there was significantly higher confirmed detection rate of ILC by fluciclovine on combined region level compared with PSMA and conventional imaging, as well as on the patient level compared with conventional imaging. There were no significant differences in conventional detection rate between conventional imaging and gallium PSMA, confirmed detection rate rather, on the patient or region level. Do you want to, I could do questions now. We have enough time. I'm about to get to that, and the answer is no. So as far as concordance, so kind of ESP, we've been working together for so long. You knew what the next slide was going to be. Fluciclovine was positive in all metastatic regions that were positive on gallium PSMA. But in 2 patients or 5 regions, fluciclovine was positive while gallium PSMA was negative. So anything that was positive on gallium PSMA, fluciclovine was also positive. And when we look at primary lesions in a subset of patients, fluciclovine was positive in all patients positive with PSMA, and fluciclovine detected 2 more primary sites of cancer in 6 out of 6 patients versus 4 out of 6 patients with gallium PSMA. Now, there is a 7th patient there, and the story behind this 7th patient is the patient had known metastasis with lobular cancer to, I think, the stomach and abdomen. No one was ever able to find the primary, and the patient had breast MRs. okay, which is very, very sensitive. And as best as maybe we could determine, maybe the patient had ILC at one point, maybe it burned itself out at the primary site, but no technique ever detected the primary lesion in this 7th patient. So it was kind of an outlier. Now, when we look at the reader agreement, it was very high. It was 90 to 100%. Slightly better with PSMA 11 than with fluciclovine, but still very high. Now, one of the reasons for detection was the fact that there was more intense uptake with fluciclovine versus PSMA 11. So if we look at comparison on SUV max on fluciclovine and gallium PSMA, and again, we're just comparing the 2 PET techniques here because conventional imaging is not going to have radio tracer involved. So if we look at the primary lesion, there's higher uptake with fluciclovine, significantly so, than PSMA. With axillary nodes, even more significantly so, with fluciclovine versus PSMA. Extra-axillary nodes, well, kind of nothing was detected with PSMA, so technically there's no statistical thing to do there. And distant mets, actually, they were about the same in terms of uptake. And if we just combine all regions, we could see in general there was a statistically significant higher uptake with fluciclovine than PSMA. If we look at the primary breast lesions, the tumor to breast, normal breast ratio for primary breast lesions identified on fluciclovine was higher than for PSMA, but this was not statistically significant. Everything in imaging is really about tumor to background and lesion to background. And this kind of goes for any type of imaging, really. You're looking at the differences in contrast, whether it's CT, MR, PET. So from this trial, this is a biopsy-confirmed ipsilateral axillary lymph node met. We could see how hot it is with fluciclovine. And with PSMA, well, kind of one of the giveaways is that the axillary node is so big, but it's really not that hot. And I've got to tell you, if the axillary node wasn't that big, with PSMA, we not infrequently have a non-specific uptake at prostate cancer and axillary nodes, inguinal nodes. It may have not been called. Here we have, and this is where it really makes a difference, IM node. Okay, so with fluciclovine, you could really see it. With PSMA, you didn't see it at all. And this is an antral metastasis. It was abnormal uptake with fluciclovine, so you could see it here. No uptake with PSMA and CT scan, you can kind of see it's very thickened. So it was called abnormal on that CT. This is bone metastasis. Now, this was biopsy confirmed in a different bone, okay? But the patient had multiple bone metastasis. But fluciclovine, we could see uptake. PSMA, there was none in this acetabular lesion. But as further proof, even though this wasn't biopsied, this patient also had a FES or estradiol scan, and this was positive there too. That's highly specific. And as we look at the primaries, one of the things we saw with PSMA is that it seemed to light up normal breast tissue. So it was just harder to see anything in that background. So fluciclovine, you're able to pick out the primary ILC lesion, which was grade 1, but there was no uptake with, or that you could discern from background with PSMA. Now, we were excited to potentially, look at SUV max compared to FDG, but as it just turned out, and FDG does not perform as well, there was no primary lesion or axillary nodal disease, which was confirmed positive on the subset of patients who had FDG. So we really couldn't compare uptake on FDG with the other 2 radio tracers in these regions. But if we look at distant mets, they were all about the same. But again, there's just very, very few here to make a meaningful comparison. But this kind of fits with some of the other data that we're seeing from this trial. So there's significantly higher uptake on fluciclovine in malignant axillary lymph nodes versus the primary breast lesions. This confirmed the observations from the earlier 2 trials. We know that there's prominent utilization of amino acid as fuel for cancer cells, And this was reported in a study of lymph node microenvironment in chronic lymphocytic leukemia. Now, again, it's not breast cancer, okay? So I'm trying to draw in from the literature all over the place. So a similar study of the nodal microenvironment of ILC may be reasonable. I think the strengths of this study were prospective clinical trial design. Each patient underwent both experimental scans as their own control. So that also demonstrated if you set up these studies correctly, the practical feasibility of this type of study. In fact, we're doing a study now with Dr. Johnny on RHPSMA for prostate cancer, where each patient is getting one scan without LASIX, one scan with LASIX. And so you're able to do with the right motivated patient, and if you arrange the times correctly, and they're not too sick, 2 scans in each patient. And where patients were compared against the composite standard of truth, histologic proof was available for most patients. Limitations, still is in R21. So we had a relatively small number of patients, limited distribution across molecular subtypes, and true negativity was not evaluated. The study was not really set up to follow these patients over a long period of time, which would enable us to look at true negativity. So what are some of the clinical implications of this possibly? Well, now there's some research going on into amino acid-directed therapies. Uptake of fluciclovine in breast cancer is primarily attributed actually to ASCT2. So fluciclovine works by actually a number of amino acid transporters, but some other research has shown that ASCT2, which transports glutamine, and fluciclovine probably is transported most like glutamine, as mostly involved. And amino acid-directed therapies may be further explored in ILC, such as dietary methionine restriction, glutaminase inhibition, and I'll get to that. And there's maybe some other tailored therapies that target amino acid transporters. And I think that we're just beginning to look at these as a scientific community. So why didn't PSMA perform as well as predicted? Well, we found that in a different study of 64 ILC patients, there are very few series with these numbers. 81% had either 0 out of 3 or 1 out of 3. This is just a tissue study, it had nothing to do with PET. Immunohistochemical PSMA expression. So I think this could explain our findings of relatively lower uptake of PSMA-11 compared with fluciclovine. And although there's no significant difference reported in vascular density between IDC and ILC, unlike IDC, angiogenesis in ILC is dependent on factors other than VEGF. And this may also be related to a lack of desmosplastic stroma in ILC, as VEGF is responsible for the expression of fibrin. So maybe some more axes to investigate. The bottom line is, for patients with ILC, novel molecular imaging of amino acid metabolism deserves further study, whether it's fluciclovine or maybe some other amino acid radiotracers that are being investigated. So our next steps are analyzing the ctDNA. So this was sent off to one of our colleagues in Harvard. And let me tell you, we complain about our bureaucracy. It's nothing compared to the Harvard bureaucracy. So hopefully they're going to get to these very soon. And so, but the world expert is there doing the ctDNA for breast cancer. So the results are pending from that. And they just emailed me a couple weeks ago, and they're saying that they're hot on this. There's more opportunities here. So now we have radiotracer called FES, or fluorestradiol. It targets ER receptor status, and it's called Siriana. And we've been using this clinically. Most ILC is ER positive. Not all are, and not all ER positive lesions, even interestingly, are shown up with FES, but this could be something to investigate. And there's also the latest kind of radiotracer flavor of the month. It's called FAPI, and everyone's going crazy about this. It's gallium-labeled fibroblast activation protein inhibitor. There may be some fluorinated versions under development, and this has demonstrated promise in the detection of ILC as well. A small subset of our patients, 3 of them, had confirmatory FES scans, and we're going to do a future analysis on that as well. So maybe next iteration of this is FES and FAPI may also be further investigated to better understand the pathophysiology of ILC. But to me, the most important thing is not necessarily finding the lesions, obviously very important, but I think what we have to do as physician scientists is examine the differences in molecular properties accounting for these detection rates. And this may lead us to further understanding the biology, which can lead us then to novel therapies. So I think the most intriguing thing here to me is this mystery to be investigated, is that both in our earlier studies and now, there seems to be higher mean uptake of cyclovine and axillary nodes than in the primary lesion. So is this a red herring, or is this a clue deserving of further study? And how should we investigate this phenomenon? Well, there's some work going on at Penn, and this is in preclinical model. So what they did was take mice, and they had triple negative breast cancer lines and also ER positive lines, not specifically ILC. And they were testing a novel glutaminase inhibitor called CB839. And they found that with flucyclovine, there was increased uptake observed after CB839 treatment in the triple negatives, exhibiting high glutaminase activity at baseline, but not in this MCF, which is ER positive, which had low glutaminase. So they also concluded that cellular uptake of flucyclovine was mediated by glutamine transporters. And so they thought that flucyclovine could have potential as a clinically translatable pharmacodynamic biomarker of this glutaminase inhibition. And this is Dave Mancroft's group up there, who many of you know. Yet, most ILC is ER positive, and they didn't see these results with the MCF, so it's kind of interesting as well. So anyway, just some food for thought. I think that there is some possibilities here, both on the preclinical and clinical studies for all those in Winship. And I'd like to thank all my collaborators, and especially a shout out to my two research associates, Eliza Mushtaq, who's sitting back there, and Ismail Lal, who's listening at home. And I consider them co-presenters of this presentation because they helped a lot throughout the whole trial, as well as helping to make this presentation. And a great shout out to all our patients who volunteered for this, and all my colleagues at Winship, who really were amazing accruers in this trial. And I can take questions.
Thank you, Dr. Schuster. If you have any questions, please use the Q&A feature located at the bottom of your screen. While we wait on questions, I want to mention that next week we have Dr. Zachary Buchwald from Emory University giving a lecture on using in situ vaccination to enhance the anti-tumor CD8 T-cell response. To view all upcoming Winship Grand Round lectures, please visit the Grand Round page on the Winship Cancer website or Winship calendar. Thank you. Any questions on Zoom? Oh, yes, Dr. Johnny.
I think you'd have to do a lot bigger study and kind of a registry trial. And so the question is, at this point, you know, does Blue Earth, you know, who developed flucyclovine, really want to put the resources into that? You know, would it be practical also using flucyclovine, or maybe we want to come up with a different amino acid radiotracer that has maybe different kinetics? Because flucyclovine, I would say the advantage, but also the disadvantage is it has fast kinetics. So you have to image like within really 5 to 15 minutes to really get your bang for the buck. But there are other amino acid transporters that are being investigated that might have different kinetics in that you can image after a certain period of time and it's stable. So from a practical viewpoint, I sort of doubt that they're going to put their resources into that. If they're going to put their resources into anything, it'll be using fluciclovine as a biomarker for brain tumors. I think that's the next thing they're looking at. But this is all very interesting, and this could lead to other spin-offs. So there's what is useful, and then there's what is practical and financially feasible. So. Yeah, typically they're looking at hundreds. Yeah. So ideally they want, so it depends. If you look at FES, there was a whole bunch of like smaller phase 2 trials. I think you would need a whole bunch more trials or a phase 3 registry. Ideally, you want a phase 3 registry trial. I mean, that's what they're really looking for. So. Yes. Mm-hmm. No, that was not on their side. That was on our side mostly. I have to say there was some delay on their side. So we were getting free radio tracer from Telix, which has developed the PSMA, and at first we're compounding it ourselves. Then it became FDA approved and commercial available, and Blue Earth, who's giving us free doses of fluciclovine. But I would say most of the contracting delay, I think, was on our side. Yeah. No, getting the sub from Hope wasn't that difficult. Yes, there's some research on that. Yeah, yeah, yeah. That I don't think we know enough, and I could tell you that as someone I once looked into trying to do a study on trying to seeing if certain dietary modifications could increase or decrease fluciclovine uptake in prostate cancer, and in the end I just couldn't get any traction on it. It turns out to be so complicated, as you know, and that the amino acids that are in our bloodstream at any one time vary, right? So, and there's so much temporal variation that, first of all, yes, I think we do know that a good diet, okay, in general, does reduce certain risks, right? And, reduces also maybe being overweight, things like that. So I think that the human body has just so much, I don't think there's a simple answer, right? My gut tells me that it's probably not just methionine restriction. It's probably a combination of factors. And I sort of doubt that you could just do dietary restriction and cure cancer that's already developed. I think it doesn't hurt, and I think it might help, but I don't think it's a magic bullet, right? So, yeah. Sure.
Microphone so they can hear.
Okay.
So Dr. Kolensky says, great work. Would a next study step be to longitudinally evaluate PSMA imaging and metastatic lobular and see if it's better than conventional imaging, given our concern that conventional imaging is underappreciates the extent of disease?
Yeah, so I think that, yeah, I think one thing we've shown in the study is that PSMA is not the way to go, okay, for at least lobular. And I would rather, look at other radio tracers, such as FES or FAPI, for staging. And then again, the amino acid metabolism, I think, is intriguing from a scientific viewpoint, and what else we could come up with. One of our colleagues at UAB is developing a different kind of amino acid radio tracer that might be interesting to look at eventually, at prostate cancer as well as breast and other cancers. So the answer is yes, conventional imaging is definitely not good. I don't think PSMA is the answer, so I think we've answered that editorial call to arms. And then now we should go down, other paths, which is, what science is all about. Right?
And Dr. Schuster, did you have many patients who had started systemic therapy and then had to wait the 4 weeks? Because by then they'd probably be done with their neoadjuvant chemo if they had to do it.
It wasn't neoadjuvant as much as patients who had metastasis who were on, various systemic therapy. So what we were able to do was sort of, if they had a pause or they're about to switch, the medical oncologists are very good about saying, let's get it in this window just before we're about to start the next one. So that worked in some patients as well. And then the patients who just got diagnosed, we were able to get them in quickly enough, before they started any therapy. So, yeah. Great. Okay. Thank you very much.
Thank you.
Thanks.